Hello, everyone. Thank you for the introduction and thank you for joining in. I'm very honored to talk about my research today on the microbial control on the ocean cycling of trace metals under the current and changing climate. And in general, I'm very interested in the interaction between microbes and chemical elements in the ocean. And what I mean by microbes here are small organisms that smaller than 1 micrometer in size, such as viruses, bacteria, phytoplankton, and small zooplankton. In the ocean, microbes play an important role in controlling the flow of chemical elements such as carbon, nitrogen, phosphorus, silica, and iron. Because, for example, phytoplankton consume and require carbon, nitrogen, phosphorus, silica, iron, and other trace metals to produce organic materials. The organic materials produced by phytoplankton can be eaten up by zooplankton and transferred up the food chain until fish and some of the organic materials produced by phytoplankton can be eaten up by bacteria or sink from the surface to the subsurface ocean and remineralized there in the subsurface ocean by bacteria. So microbes play an important role in controlling the ocean cycling of chemical elements, and vice versa, chemical elements can control the growth of microbes in the ocean. So that's why I'm interested in the interaction between chemical elements and microbes in the ocean. And that's why over the last few decades, oceanographers have been trying to understand the processes that control the cycling of chemical elements and the distributions of chemical elements in the ocean, and now have a decent understanding of the distributions and the processes that control the cycling of nutrients like phosphate and nitrate in the ocean, as shown here in the map that showing the surface distribution of phosphate in the ocean from the World Ocean Atlas 2018 data set. And you see that we have high concentration of phosphate in the Southern Ocean, subtropical Pacific and North Pacific, and low concentration of phosphate in the regions such as Indian Ocean, subtropical giants of the Pacific and Atlantic Ocean, and those regions are where phosphate can be a limiting factor for the growth of phytoplankton and microbes. And also we have a decent understanding of the stoichiometry, the content of nitrogen and phosphorus inside the cell of phytoplankton and microbes. And also we have a decent understanding of the ratios of carbon, nitrogen, phosphorus in the ocean and inside the cells of phytoplankton and microbes in general, as proposed by an American oceanographer in 1950, Redfield. He proposed that the ratios of carbon, nitrogen, and phosphorus in the ocean and inside the cell of phytoplankton and microbes is relatively constant between 106, 16, and 1. But recently, researchers from University of California, Irvine, suggested that these ratios can be deviated from the Redfield ratio as a function of temperature and environmental conditions in general. But in general, if you look at the big picture at the global scale, the Redfield ratio still holds true. As we're looking at the figure on the left here, we see the relationship on the right here, the relationship between phosphate and nitrate is still 16 to 1, is still a linear relationship in the ocean concentration of nitrate and phosphate, but also inside the cells of phytoplankton and microbial cells. And also on the figure on the right here, we see that nitrate tends to be depleted before the depletion of phosphate, and in general in the ocean, nitrate is a limiting factor for the growth of phytoplankton and microbe. But we can also say the same thing for trace metals in general and iron in particular, the most important trace metal in the ocean, because it's very difficult to measure trace metals in the ocean. And as of 1997, we only had few measurements of iron concentrations in the North Pacific, in the North Atlantic, and some regions in the Southern Ocean. Thanks to the improvement in the measurement techniques, recently we have much more data of the iron concentrations in the ocean coming from cruises that are going around the ocean, but still we do not have the global map distribution of iron in the ocean, even in the surface, and not to mention for the subsurface ocean, as we have for phosphate and nitrate. And also we do not know about the stoichiometry, the iron content of inside the phytoplankton cell and microbe cells. And I would like to emphasize that we know very little about the metal microbe interaction compared to what we know about the microbe and phosphorus and nitrogen interaction. And that's gonna be the main research questions of my research activities. In particular, I would like to ask three general questions. Question one, how do microbial processes control ocean ion cycling? I'm gonna focus on iron because again, iron is the most important trace metal in the ocean. And I would like to know the relative contribution of the microbial processes control on ocean ion cycling versus external sources of putting iron into the ocean, for example, from atmospheric deposition. And second, how do trace metals, but specifically iron, affect the growth of microbes in the ocean? And what's going to be the consequence of the microbial competition for iron? For example, the competition for iron between bacteria and phytoplankton. And finally, how is the microbe ion induction affected by climate change and human perturbations? And to answer these scientific questions, I'm gonna use ocean biochemical model, and I'm gonna focusing on global scale and large scale processes. And what the model is trying to do is a model trying to represent different components of the marine ecosystem and ocean biochemistry. For example, the model trying to represent different types of zooplankton and different types of phytoplankton and different chemical elements. And that is shown on the figure on the right, on the left here, the conceptual figure showing how we visualize the ocean biochemical model. And on the right hand side is that we trying, what it's showing is that we're trying to translate the conceptual understanding to mathematical equation to know the biochemical state of the ocean at a particular time of interest. For example, here we know that there are many processes that can control the state of ion concentration at a particular time of interest and that in a particular specific location. And by using ocean biochemical model, first I'm looking at how do microbial processes control ocean ion cycling. I'm showing here first the ion concentration along the Indian Ocean. The first panel here showing observations. So we have x-axis is latitude and y-axis is depth. The second and third panels are results from the ocean biochemistry model from different model setups. The second panel is a model when I consider only I do not consider microbial processes, and the third panel is the result of the model when I consider microbial processes. And what I mean by microbial processes here are the processes controlled by microbes, controlling the production of organic ligands that protect iron from scavenging, and the microbial processes that control the particulate production and remineralization of particulate iron. And we see that if we do not consider microbial processes, the iron concentration in the ocean tends to be uniform and cannot reproduce what you see in the observation, the sharp gradients between the north and the south, and the sharp gradients between the subsurface and the surface ocean that we see in the observations. And to reproduce this sharp gradients, we have to consider the microbial processes as shown here on the third panel. So I conclude that microbial processes control the ocean ion pattern and variability by controlling the organic ligands production and the remineralization and production of particulate ion. And that is very important because if you look at the response of the marine ecosystem here in the Indian Ocean to increasing ion deposition, we see that when you do not consider the microfeedback, the change in net primary production is increasing everywhere almost everywhere in the Indian Ocean. But when you consider the microbial feedback, the change in net primary production is much smaller and much more diverse. We have regions with increasing NPP and regions with decreasing NPP. So I conclude that the microbial control on ocean ion cycling modulate the marine ecosystem responses to human perturbations. And another aspect of how do microbial processes control ocean ion cycling can come from small zooplankton. We see that when we have high ion requirement for small zooplankton, the surface dissolved ion concentration can significantly decrease everywhere in the ocean, as shown here by the blue color by the global map here. So I conclude that the ocean ion concentrations are very sensitive to the ion demand of small zooplankton. And the second question I would like to ask to answer is how does iron affect the growth of microbes in the ocean? And it turns out that microbial growth, bacteria and small zooplankton is very sensitive to iron in low iron regions such as the Southern Ocean, the tropical Pacific and the North Pacific, as shown here by both figures on the left and on the right, showing that when you have high iron requirement for bacteria and high iron requirement for small zooplankton, their biomass can significantly decrease in low iron regions. And what is going to be the consequence of the microbial competition for iron? That obviously is a competition between bacteria and phytoplankton for iron, as shown here on the map, showing the global map, showing the contribution of bacteria to the total biological iron uptake in the surface ocean. We see that bacteria can contribute to around 30 to 60% of the total biological uptake in the upper ocean. And when we have high iron storage for iron for bacteria, they can decrease the magnitude of the seasonal cycle of phytoplankton bloom in the oceans, especially in the Southern Ocean, the tropical Pacific, North Pacific, and North Atlantic. So we conclude that active competition for iron between microbes can counter the phytoplankton blooms in iron-limiting regions. And for future research, we see that I do not answer yet the question about how is the microbe interaction affected by climate change and human perturbations. And we just see that the microbe ion interaction can change the response of the marine ecosystem to human perturbations, and also how are the interactions between microbes and other trace metals play out, how these interactions will change in the future, because we know that iron is not the only trace metal that can play a role in the growth and activity of microbes. Cobalt, zinc, copper, and manganese, they all can play a role in the activities and growth of microbes in the ocean. And also, what is the role of viruses? We know that viruses can kill bacteria, zooplankton, and phytoplankton, and of course, they're going to play a role in the flow of carbon and chemical elements in the ocean, and play a role in the interaction between microbes and metals. And for the impact of climate change and human perturbations, we know that trace metal coming from the atmosphere is increasing because of the human activities. So what is gonna be the impact of that on the metal microbe interactions? Also, the ocean is warming up, turning soil, that is, they become more acidic, and they're losing oxygen, so they're losing breath. All of these processes is a function of how we're gonna emit CO2 in the future, and all these processes gonna play a role in the metal microbe interactions, because they change the microbial growth activity and trace metal requirements. And we can form several simple hypotheses. For example, we know that increasing iron deposition into the ocean increasing bacterial activity, and bacteria can produce ligands to protect iron from being removed from the ocean. So we can have a positive feedback loop between bacteria, ligand production, and iron concentration. Another hypothesis that we can form is the warming effect on bacterial activity. The ocean becoming warmer, bacterial activity is increasing. The activity of bacteria living inside particle is increasing. So we have more export of particulate matter from the surface and subsurface ocean. So the surface ocean can become more severely limited by metals. And this hypothesis and other hypotheses really need to be addressed in future research by using ocean biogeochemistry model. And with that, I would like to conclude my talk and happy to take any question. Thank you so much.